Hello everyone, and welcome to Chapter 27, which is on water, electrolytes, and acid-base balance. So like I said, this chapter is only 19 or 20 slides, so I'm going to try to keep it brief, but I will give you a little bit of detail that will help you in your future nursing classes. So this is the um, let's say the bread and butter of what you need to know here for chapter 27. And like I said, use your study guide. That's what is going to be focused on for your exam, everything that has its text in black. So let's start with osmolality. We kind of talked about this in the urinary system. Osmolality is going to be the measure of water versus the solute concentration. The higher the solute concentration means the higher the osmolality. So I really liked the image here on the right hand side where it shows you a drop of water, and you can see the drop, sorry about that. You could see within that water we have these little dots representing solutes. And so this one over here would have a higher solute concentration compared to the one on the right. If we add or remove water, that's also going to change the osmolality. So I did give you an example in your study guide where and I just made these numbers up, we have water that is 5 milliliters and contains 1,000 salt granules. So we're going to say that has high osmolality. So we could just picture this guy here to the left, drawing all over here. And then if we were to take that and remove 2 milliliters, so now we have a solution that's 3 milliliters containing 1,000 granules of salt still, that means that we have an even higher osmolality. Now if we took that same solution and added water to it, so in the example, I think I added 3 milliliters of water, and so as a total we have 8 milliliters, and still our 1,000 salt granules, that would mean that our solution B, let's say, has a lower osmolality now. So changing up these osmolalities are going to change the mechanisms within our body to keep our homeostasis. Before we get into increase and decrease osmolality, I just wanted to touch upon what these body fluids are and how they're divided up. So I'm just going to kind of draw a little picture for you here so you can kind of place this in your mind. So if we have a cell here, okay, this is my nucleus and my beautiful rough ER, my mitochondria here. Okay, so there's my cell. We're going to call that, anything in there, we're going to call it the intracellular fluid. Okay, so we'll just write that in here, intracellular fluid. Sorry, I don't know why my handwriting looks so jagged. So all of our intracellular fluid, that is going to make up about 40% of our body weight. And how much fluid, how much water really is found in our body? Anyone know? I know you all can't answer me, but within the body, we have water. I'm just going to write this off to the side. Water making up 60% of our body weight. That's huge, right? So all these changes are really important. Okay, so intracellular is 40%. That is about 2/3 of our total body fluid. And what's going to be our other 1/3 that's going to be left is everything outside of the cell. So that is going to be called our extracellular fluid, which we've already mentioned these terms many times before in class, right? Extracellular. And so extracellular, we said that is going to be the remaining 20%, of our full 60% water from our body weight. So this would be, extracellular would also make up that 1/3 of our body weight. And by the way, this is the area that is prone to loss. So if we're losing water, it's almost always coming extracellularly. The way this is broken down is we find it within our interstitial fluids, which, as a reminder, this is, oh geez, this pen is really acting up. Interstitial, which this is the tissues, right? This is the tissues outside of our cell. And this is going to be the biggest part of our extracellular fluid. This is going to make up 80% of our extracellular fluid. The other thing that plays a big part in this is intravascular fluid. I apologize for this. I'm going to try and pause in a minute and fix this. And that's going to make up about 20%. And we also have one more, but it's kind of a negligible amount. This is called your transcellular fluid. And what that means, I won't write it out since we're having issues here. What that means, it's the fluid that we kind of find in special compartments. So like the cerebrospinal fluid within your brain or synovial fluid that you find within the joints. And how are we going to lose fluid from this area? It is going to be through your urine, through evaporation. So like if you go and work out and you sweat, so that sweat that's evaporating off of your body. It could be from your respiratory passages as well. You know, so you have moisture when you breathe out. And then also through your feces. So now that we've kind of got where these fluids are coming from, let's talk about what happens when we increase the osmolality or decrease the osmolality within them. So if we have increased osmolality, that means that our body is going to trigger thirst from the hypothalamus and start to secrete ADH, the antidiuretic hormone. By the way, I think I forgot to mention this in their urinary system. Just so you know, this is also called vasopressin, and we'll talk about that more in our endocrine lecture next quarter. And then for decreased osmolality, of course, we're going to inhibit our thirst and also inhibit our ADH secretion. So what's really going on here is if we have increased osmolality, we are creating a hypertonic fluid. And that means we have increased the concentration of our solute particles greater than our intracellular fluid. So what happens to that cell? If you remember from biology or chapter 3, it means that the cell is going to shrink. So it's almost kind of like the cell is dehydrated. So you can think dehydrated, we're going to get thirsty, right? Okay, so then with decreased osmolality, we are creating a hypotonic fluid and we are going to decrease the solute particles within our extracellular fluid compared to that of our intracellular fluid. And now we are going to have a cell that is going to swell. So we have a shift of the fluid from our extracellular fluids into our intracellular, I should say compartment, into our intracellular compartment. Okay. This reason, increased osmolality really with the cell shrinking and dehydrated, is really the reason also that we cannot drink salt water. If you drink salt water, you will die because it will pull that water into our gastrointestinal system, and our system will basically excrete all that and our cells will dehydrate and cause us to die. So let's move into thirst. So what's going on here? With our thirst, we have within our heart our baroreceptors. Do you remember what baroreceptors measure? They are going to measure pressure. And so when they sense a change in pressure, like what do they give in this example? Reduced blood pressure. Then it's going to go over to our special friend, the hypothalamus. Oop, clicked in there. I just wanted to kind of wave in there. I don't know if you guys can see my mouse, so I'm just going to kind of color this in. But your hypothalamus is right over here. I'm going to color it in red. Our hypothalamus. That is going to be the area where we are going to have our osmoreceptors, okay, which osmo for osmolality, right, are going to be triggered, and we want to increase osmolality. And we're not only getting input from our baroreceptors within our heart, and we already talked about this in cardiovascular system. You're going to find it within the carotid sinus and aortic sinuses, right, within the aortic arch and the carotid artery. And we're also going to get triggers from our juxtaglomerular apparatus in the kidney. So that tells our hypothalamus we need to increase osmolality, like I said, and we increase our thirst. That causes you to drink more water, and as a response, our blood osmolality is going to go up, and we are, our blood pressure is going to go up, and we are back to homeostasis. And this is just another image discussing a shift in blood osmolality. So I mentioned to you in class that our blood osmolality, we talk about it as if it's 300 milliosmoles, but the actual number is going to be, I think I said 270 in class. That's like a normal, close to the normal range, but it should be around 275 to 290 milliosmoles per liter. Okay, that's a normal blood osmolality range that we want to stick to. So we usually use 300 for simplicity. But anyway, if we are at homeostasis and then we start to see that we get an increase in blood osmolality, okay, too many solutes within our plasma, within our blood, then we are going to trigger that hypothalamus, the osmoreceptors within the hypothalamus. We see that we have an increase in blood osmolality, and we secrete ADH, yes. And that's going to cause thirst. Do you guys remember what ADH does? Well, it says right here, but we're going to go over to our distal convoluted tubule and our collecting duct. And remember that what that does, having ADH there, is it puts in those aquaporins, right? And aquaporins do what? They allow for H2O to move out the channel and be reabsorbed. We reabsorb the water back into our blood. And so that's what ends up happening. Our water is going to increase, and that brings our blood osmolality back to homeostasis. Of course, the opposite can happen too. Our blood osmolality is going to decrease here, and so our osmoreceptors in our hypothalamus again is going to detect this and tell our posterior pituitary to either reduce or stop ADH, and that's going to decrease our thirst level. And so our body will not be able to get rid of the water that we have within our filtrate, right? So it's just going to stay there. So we get rid of these aquaporins. They're going to disappear. And so the water has to stay in here. And so that's going to be excreted as urine. Okay, and that helps to bring our blood osmolality back to its homeostasis level. Hey guys, so before we go into the regulation of extracellular fluid osmolality, I threw in this extra slide. You don't have it, but you can write it into your notes. I wanted you to have an idea, an overall picture of how we regulate our extracellular fluid volume. So it's going to be through two ways. It's going to be through our neural mechanisms, or it could be through hormonal mechanisms. And you can kind of notice some of the names down here that you are already familiar with. So that's kind of nice, right? Less to study. So with our neural mechanisms, it's going to be our sympathetic nervous system that really assists us in doing this. And remember, our sympathetic nervous system is your fight or flight response. So we have a certain frequency of action potentials that are sent through our sympathetic neurons to those afferent arterioles of our kidneys so that we can sense if there's any changes in our blood pressure. So what can happen is our baroreceptors within our arterial and venous blood pressure, if that is going to, let's say, increase, okay, we have an increase in our blood pressure we're sensing, then that means that we are going to actually have a decrease in the frequency of our sympathetic fiber action potentials to our afferent arterioles that we would find in our kidneys. And so what that would cause is a dilation of these afferent arterioles. What happens when we dilate arterioles? It means that we get more blood moving through, right? So we are going to then in turn have an increase of our GFR, our glomerular filtration rate, which will increase our filtration volume, or filtrate, and that will result in increasing urinary volume. Okay, so you're going to have more urine output. And vice versa. So we can switch these arrows around and the opposite will happen. Now for our hormonal mechanisms, I'm not going to go through these mechanisms again because we talked about them in our urinary system, and I hope you've got these down. If not, I want you to take a look at figure 27.4. I'll put an F for figure. That gives a really nice summary of these 3 hormonal mechanisms. And also you could take a look at table 27.5 in your book as well. That's a nice table that summarizes everything. The one thing I do want to mention is, when do these hormonal mechanisms work? So with your renin, angiotensin, and aldosterone hormonal mechanism, this is sensing for more of small changes. Put small. I'm going to use my triangle for changes. Small changes in blood volume. Okay, so these are kind of those micromanagers you can think of. And then our atrial natriuretic hormone, that is going to be in charge of more large-scale changes. Large change in blood volume. And that makes sense, right? Because our atria have to feel the change within their atrial wall, the stretch within their atrial wall, in order to know, hey, there is an increase in blood volume, okay? And they're not as sensitive as feeling a decrease in blood volume because of that reason, because of that mechanism there. And then so last one that's left is ADH. We've talked extensively about this too. It is going to help to regulate our extracellular volume through large changes in blood pressure. Okay, and these are going to be changes of like 5 to 10% in our blood pressure. All right. Oh, and one more thing before we move on into our electrolytes. I just quickly wanted to mention that the regulation of our intracellular fluid is going to be different than this extracellular fluid volume because we have different components at play. Think of all the setup inside of the cell, but one of the main players is our plasma membrane. We have a bunch of proteins there that are going to help ions and other substances move in and out of the cell, but also water can move easily across through osmosis into and out of our cell. So that is going to, the fluidity of that, the regulation of it is going to be determined by changing the concentration of our solutes in our extracellular fluid and our intracellular fluid. So think back to when we have our hypotonic and hypertonic solutions and what that does to our cell. Okay. All right. Oh, I never went through the slide. Why am I switching? Okay, so that brings us to our electrolytes. Our electrolytes are going to be molecules or ions. Usually they are going to be the ions with an electric charge. Now, why that's important is because they are going to play a key role throughout our body in generating electric currents. So, for example, action potentials through neurons. Ingestion is going to add electrolytes to our body, so through our digestive system, and then our kidneys, liver, skin, and lungs are going to work to remove them from our body. And of course, our concentration is going to change when we're growing or gaining and losing weight. But I wanted to draw a little image for you to keep in the back of your head as you move through your anatomy courses here. So this is my cell. Ooh, it's a little wobbly cell, but anyway, what we're going to have the most of in here, the most electrolyte or ion really, will be potassium inside the cell. We still have other ions like a little bit of chlorine. We have some sodium moving in. You have phosphate. This is going to be kind of the key anion in here. We have a little bit of calcium, some magnesium, and did I miss anything? I think I got them all. So that's kind of the key players and little sidekicks, let's say, going on inside the cell. Outside of the cell, we've already talked about this. The main player out here is sodium. Right? Sodium. And the other things that we can expect to find here are a little bit of potassium, chlorine. We also have calcium. I think I got them all. Yes. So again, big ones to know. Potassium, huge on the inside. Sodium, huge on the outside. So keep that in mind as we're moving through. I say this right here. Sodium ions are dominant in the extracellular fluid. We see it right here. And they're responsible for 90 to 95% of our osmotic pressure. So they're kind of setting the bar right there. So how do we regulate sodium? We do it mainly through our kidneys. We just talked all about this, right? And small quantities are going to be lost in sweat. So either that's going to be through insensible perspiration, so water just evaporating from the skin, or sensible perspiration where it's secreted by our actual sweat glands that, oh, it got rid of my picture. Anyway, that is secreted by our sweat glands. So we're going to have our solutes in there. So the next thing we're going to talk about is hypernatremia and hyponatremia. So hypernatremia is when we have elevated plasma sodium, meaning we have too much sodium within our blood. And hyponatremia is going to be when we have a decrease in sodium within our blood. So actually, before we jump into this on this next slide, I want to quickly give you an idea of what each of the major electrolytes kind of do in the body, just again, so you have it in the back of your head, and all of this kind of makes a little bit more sense as we're moving on. So let me get a blank, yeah, I don't have enough room here. Let me get a blank slide and we're going to write this out. Let's see. Oops. Okay. So let's start, and hopefully my pen cooperates with this. So let's start with sodium, since we just talked about it. So with sodium, we said this is a huge player in the extracellular matrix. So we're just going to write extracellular excitation. Like, it's going to help with creating action potentials within nerves and also our muscles and some other functions within the body. Next, let's talk about potassium, since we mentioned that as a key player inside the cell. So we're going to say that this has intracellular excitation as its main function. Okay. And then let's move to magnesium. Okay, magnesium can be used for muscle relaxation. Just going to abbreviate here. You can actually, if you suffer from insomnia, you could take some magnesium and it'll just help your muscles kind of relax and allow you to sleep a little bit better too. Next, let's move to phosphate or phosphorus. This is going to assist us in muscle contraction. So kind of the opposite of magnesium here. And since we're talking about muscle contraction, let's talk about calcium. Most people think calcium and they think of bones, and that is correct. Of course, they're going to be in our bones, but they're also going to assist us in muscle excitation. Okay, if we don't have calcium, we cannot contract our muscle. Keep that in mind for when we get into chapter 9. And the last electrolyte I want to talk about is chlorine, and this is also going to help us with some cellular excitation. Okay, you got that with my hieroglyphic writing? Okay, so anyway, let's move into our different types of sodium in the blood issues. So at the very top we have hyponatremia, and so we said that means we have too little blood sodium. So either we're not getting enough sodium within our diet, which usually that's not an issue here in America. We can have extra renal losses. Maybe we're putting too much sodium within the urine for whatever reason, dilution, or hyperglycemia. And if you look at these symptoms here, we're going to see that a lot of these have to do with neural issues, because we said that sodium helps kind of send action potentials through our neurons. So lethargy, confusion, apprehension, seizures, and coma, that's all related to nerve issues. So then it says, when accompanied by reduced blood volume, we have reduced blood pressure, tachycardia, and decreased urine output. That makes sense because we are having our kidneys trying to keep our sodium within our blood, and so we want to keep our water as well. And so we are going to see that we have decreased urine output because we're trying to keep in that water. And then when accompanied with increased blood volume, we can have weight gain, edema, and distension of veins. So it's kind of pulling in the water and salt back into your body and maybe into the tissues, and that's how you're going to get your edema there. So next, let's move into hypernatremia. This could be from high dietary sodium, which is more likely easily to occur when it's a dietary issue. Administration of hypertonic saline solutions or oversecretion of aldosterone, and water loss. So what this causes is going to be thirst, fever, dry mucous membranes, and restlessness. So this is kind of strange as far as thirst, because you're like, okay, I have all this salt, it should be pulling in water into my body. Why would I be thirsty? Well, the reason is, if you think about it, like, you have all this salt in there, but then you're pulling in all your water for that salt, and then your body's saying like, wait, I need more water because I have so much salt in here. So that's why you get thirst as one of the symptoms. Of course, you aren't using your water that you do have in your body properly, so you're going to have dry mucous membranes. It'll also cause you to have like dry and kind of flushed skin as well. More seriously, we'll have convulsions and pulmonary edema, again, pulling that water into the wrong areas. When it's occurring with increased water volume, then we start to see the same things we saw up here. So weight gain, edema, but then we'll also see elevated blood pressure and a bounding pulse. So now we are going to move into our potassium ions. Okay, so now with our potassium ions, this is maintained at a very narrow range. It is going to be at 3.9. Where's my little... Okay, I guess it's not going to let me write anymore. A lot of technical issues this screencast. Anyway, it'll be between 3.9 to 9.2 millimoles per liter compared to sodium, which has a much broader range. It could be anywhere from 9.8 to 77.2 millimoles per liter. You don't have to know those numbers. I'm just giving you an idea of where they should be. So, and the big thing, we've already kind of talked about this, is potassium is that main key player inside the cell. So it is known to set that resting membrane potential that is around negative 70 millivolts. And what we're going to see is aldosterone is going to increase the amount of potassium that we are going to secrete into our urine. So we are getting rid of it when aldosterone is present. So we have our terms hyperkalemia. This refers to having too high of a concentration of potassium within your extracellular fluid, and hypokalemia means that we have too little potassium in our extracellular fluid. So where do I have? Oh, I have this chart here, but what I'm going to do is kind of incorporate it into this regulation within our extracellular fluid. So we see that our blood potassium is at its normal range, and then let's go with increase first. We have homeostasis is disrupted, and we are going to see that we have too much potassium within our blood. What do we need to do to get rid of it? We call upon our adrenal cortex to secrete aldosterone. And so when it does that, we're going to go over to our kidney in the distal convoluted tubules and collecting ducts to tell it to use its sodium-potassium pump to reabsorb sodium, but we are going to take that potassium and excrete it within our urine, and that will help bring those levels back down so we can reach homeostasis again. So what happens when we have that increase in potassium? It means that our cell has so much potassium that we are going to reach a cellular depolarization. And so we have more excitability taking place there. And what that results in is having a lot of issues with the heart, and also a lot of neurological issues. So the mild symptoms would be increased neuromuscular irritability. And again, these are all coming from the chart that I have on the next slide. We will also have intestinal cramping and diarrhea because our enteric plexus, sorry, took me a minute, is disrupted, and that's what's causing the cramping and diarrhea. We have rapid cardiac repolarization as well. So remember, repolarization is bringing that action potential back down too rapidly, okay, going back to that resting membrane potential after our depolarization. And then if it's really severe, we're going to have muscle weakness, loss of muscle tone and paralysis, and reduced rate of cardiac action potential conduction. Now, if we have the opposite happen, we are going to decrease our aldosterone secretion from that adrenal cortex in our adrenal gland. Remember, this is the party hat of our kidney. And then we stop or decrease the aldosterone within our distal convoluted tubule and collecting duct, and that causes it to kind of salvage the potassium that we have, and our homeostasis level is returned back. So what's happening when we are in hypokalemia? It means that we are going to decrease our extracellular potassium, and that causes cell hyperpolarization, meaning our resting membrane potential within our cell was negative 70 millivolts, right? So we're decreasing that number. Let's say we go to negative 90 millivolts. Well, that's going to make it harder for that cell to become depolarized or excited, whatever type of cell it is. Let's say it is a cardiac muscle cell. If that is hyperpolarized, then that means it's a lot harder for that cardiac muscle to contract. So what we can end up with is bradycardia, or a decreased ventricular or delayed ventricular depolarization, or atrioventricular block. So we're affecting that AV node that we talked about during electrical impulse through the heart. And we will see in other areas where we have smooth muscle, we'll have more of a decreased smooth muscle tone, because again, it takes a lot more stimulation to get that muscle to contract. And the ways that we can have hypokalemia is obviously reduced potassium intake, but we can also have maybe too much aldosterone is being secreted, and so we get more of a renal loss where we get too much potassium leaving through our urine. We have alkalosis as well, which we'll get into a little bit at the end of this lecture. And insulin administration, this is still a little unknown. So, but we do know that it helps to cause hypokalemia. So I'm going to pass right through this slide. This brings you over to our comparison of strong and weak acids here. So you guys are going to be talking about this, I believe, next quarter. So if you have a very strong acid, that means we're going to have complete dissociation of these elements here. So we have our hydrogen ion on its own, and then we have our chloride ion on its own as well. So this looks more of an I, but it should be an L. Anyway, if we have a strong base, this means we have complete dissociation here too of our hydroxide ion. So our sodium ion is on its own, hydroxide ion is on its own. If we have a weak acid, which this is what we have in our body, then that means that we have only partial dissociation. So it means that they kind of live within an equilibrium, shifting either to the left of the equation or to the right of the equation based on the needs of our body. So if we have carbonic acid, we can take off of one of those hydrogens and then leave our, we're left with our bicarbonate ion here. So just some definitions here. We've discussed this in the past, but let's refresh our memory. Acids are always going to release a hydrogen into solution. When we do that, we can think of our pH scale. Remember, that goes from, let's just say 1 to 14. And so the more hydrogens that we're going to release means that we are going to go to a lower number on the pH scale. So let's say you have a solution that is at a 6, and then you add more hydrogens to it and it goes down to a 5. Okay. If we have a base, that means we are removing hydrogens from that solution. So we take out some hydrogens and we go from that 6 to, let's say, a 8. Okay. And acids and bases are going to be grouped whether they are strong or weak, just how we saw on the previous slide. Now we have some buffers in the body, and we'll talk about this in a moment, but what buffers do is they resist any changes in our pH, so that when we do add hydrogen, our buffer is going to be able to remove it. If we have hydrogen removed, then our buffer is going to replace it. So I'm going to take you back to this right over here. Like we said, if we have a base, then we could just add a hydrogen to it and shift to this carbonic acid. If we're starting to see like, oh, we want to get rid of some hydrogens, then we just shift and we take off one of those hydrogens and we're left with the bicarbonate ion. So the types of buffers that we're going to have within our body is the carbonic acid bicarbonate, which you saw in the previous slide. We also have the protein buffer and also phosphate. So here is a nice little summary of the regulation of acid-base balance. Okay, so these next several slides I'm going to kind of summarize first. So I'm going to summarize on this slide first. This is slide, well, I think for you guys it's 14. So the ways that we can regulate acids and bases within our body is to use 3 different systems. And these are listed as far as how quickly we can use them as to balance our acids and bases. So the whole point of this is to balance our blood pH. Blood pH is going to range from 7.38 to 7.42. Now this number always ranges depending on what textbook you're reading or what handbook you're reading, but I believe for all of nursing in our curricula, we kind of go with this number, so focus on that one. So the first thing we're going to do is use a buffer, and this is going to be basically that equation that we saw in the previous slides, except I'm going to add CO2 plus H2O, and then I'll do my double arrows here, our carbonic acid, and double arrows again, so this can dissociate into, or partially dissociate into our hydrogen and our bicarbonate ion. Okay, so it's using this buffer that's within our blood. So if we see that our pH is going to rise, so it turns more basic, then our buffer is going to bind more of those hydrogen ions in order to do this. And it kind of shifts everything over to the left. If our pH is going to fall, so we become too basic, then we will shift this equation to the right and we want to release these hydrogens. So we're going to talk about these hydrogens a little bit more in the coming slides. So I just want to state here that this bicarbonate ion is our great buffer. Like we said, this buffer is going to be the fastest for our body, but it can easily be overwhelmed and we would have to resort to our respiratory center or over to our kidneys to avoid acidosis or alkalosis, and we'll talk about that at the very end. So next let's discuss the respiratory center. So with our respiratory center, I know I have this text here and that is correct, but let's talk about what happens if pH rises or falls. So with our respiratory center, if pH is going to increase, what our body wants to do, maybe I shouldn't use an arrow, we'll just put a dash. What our body wants to do next is hyperventilate. So we're going to see, did I say hyper or hypo? I mean hypoventilate. So what that means is we're going to decrease our respiratory rate, and that is going to cause a rise in our blood, oh, there goes my pen again, blood carbon dioxide. So when we get that rise in blood carbon dioxide, okay, that is going to cause a decrease in our pH. Okay, if we have the exact opposite happen, pH is decreased, then we are going to hyperventilate and increase our respiratory rate. What that does is decrease our blood carbon dioxide, and that causes the shift to increase our pH to go back to our normal range. Okay, If you can talk yourself through this, you are good to understanding how our respiratory center is going to control our acid-base balance. Now, when it comes to our kidneys, okay, if the pH rises, we are going to see our distal convoluted tubule decrease our hydrogen secretion into the urine and decrease bicarbonate absorption into our blood. So we'll have more of our carbonic acid dissociating into our hydrogen and our bicarbonate ion. And vice versa. If our pH is going to fall, our DCT is going to increase our hydrogen secretion into our urine and increase our bicarbonate absorption into the blood. Now, to discuss this, I'm going to jump to, we already talked about all this, so I'm going to jump past respiratory because we just discussed it. Do we need this? I'm going to talk about this on the next slide, this one here. Okay, so let's talk through this from the beginning. So at number 1 here, we have our blood pH is going to decrease. So we have our hydrogen combining with bicarbonate ion, and that's shifting everything over to the left. So we get our carbonic acid that gets shifted into our carbon dioxide and water, and that diffuses easily into these kidney tubules. So I want you to imagine that you are in, which one are we going to talk about first? That we are in your proximal convoluted tubule, actually. So this is happening at the PCT. And so our carbon dioxide is going to shift into through this equation again. So carbon dioxide will combine with the water, we dissociate, blah, blah, blah. We get to our hydrogen and our bicarbonate ion. So what happens after that is within our kidney tubule, and I'm just going to add our lines here. Remember we said we have that cuboidal cell, so I'm going to kind of make this a cuboidal cell. Okay, and this is the lumen of our kidney. So this is where we're producing urine. I really shouldn't put urine. Let's put filtrate. Filtrate, right? And so we have special proteins or transporters that lie within the cell, right? So we're going to have a special, this would be an antiporter. I don't know why they don't have it in green here because they have the symbol here. But that takes one of those hydrogens and it puts it into our filtrate to get rid of it, right? That's what we want to do when we have our pH decreasing. We need to get rid of these hydrogens. We don't want to become too acidic. And what we exchange for that is a sodium ion. And so we need another transporter. So here's my other transporter. This is a symporter now, so we can move 2 molecules or ions across at the same time. And what it does is it moves that sodium, diffuses it into the interstitial fluid, and then back into our blood. And it takes that bicarbonate ion that was left over here and shifts that back into the blood too. What's this bicarbonate ion going to do? It's going to find another hydrogen to bind to. And so with that, it's going to decrease the hydrogen concentration within our blood, which will increase the blood pH. Okay. The last thing we are going to talk about is acidosis and alkalosis. So acidosis is when we have, let me get my pen back out. Acidosis is when we are going to have our pH body fluids below 7.35. I know I gave you a different number. I gave you 7.38, but just know it's kind of within that range. And what we want to avoid is going down to 7. If we go down to 7, we are going to die. Okay? So we have two different types of acidosis. We have respiratory and metabolic. Respiratory acidosis, we're not getting enough ventilation taking place. We are reducing our carbon dioxide. We get asthma, damage to our respiratory center in our brain, and emphysema. Remember, that's when we have, here's our healthy, nice alveoli. Emphysema kind of pulls it out a lot bigger, right? Less surface area, less exchange of gases. With metabolic acidosis, we are getting this from all conditions other than respiratory that's going to decrease pH within our body. So if you have diarrhea, vomiting, ingesting an overdose of aspirin, untreated diabetes mellitus, or anaerobic respiration. With alkalosis, we want to avoid our pH going to above 7.45. So we want to avoid going to 7.7 or above. If that happens, again, we die. All right? So respiratory alkalosis is going to be caused by too much hyperventilation where we can't control it anymore, going to high altitude areas where we have reduced partial pressure of oxygen. Metabolic alkalosis will result from all conditions other than respiratory that are going to increase pH. So vomiting, too much aldosterone, ingestion of substances like bicarbonate of soda. So the way we are going to try and control this within our kidneys at least is by using our intercalated discs. Do you guys remember where these are? Discs. Oh, I'm saying discs. Sorry, intercalated cells. If you said intercalated discs within the cardiac muscle cells, you are correct. But intercalated cells, where do we find those? Within the DCT and CT, right? Distal convoluted tubule and collecting tubules. So we said we have actually types A and B. And so type A actually helps with acidosis. So I'm going to put a big A here. And type B is going to help with alkalosis. So here's my cuboidal cells here. And what's cool about these cells is they are going to work differently depending on if you're approaching acidosis or have acidosis or alkalosis. So type A is going to work, and I'm just going to write these out. We're going to reabsorb bicarbonate ion and potassium, and we are going to secrete our hydrogen, right? And then with an alkalosis, we're going to do the exact opposite. So because we're not really working well here with this pen, we're going to reabsorb our hydrogen, and I'm going to put an S for secretion. We're going to secrete our potassium and also our bicarbonate ion. So how do we do that? I'm not going to get into all the differences here, but just know that we have these different transporters, whether they're facing the lumen or our blood supply. They are switching in and out the, let's say over here would be our hydrogen that's being secreted into the lumen of our tubules, and we're taking in potassium. So that's why we're saying reabsorb potassium here, because we're switching this up. We want to get rid of these hydrogens. And then on this side here, again, you guys don't have to know this detail, but I'm just trying to have it make sense in your head. We would reabsorb our bicarbonate ion, but we're pulling in an ion of chloride. And so if we look at our type B cells, they're going to do the same thing, but they just do it opposite. So here's my transporter, And I am pulling out the bicarb ion here to secrete it and pulling in the chlorine. Oh geez, this is looking really messy. And then the last one I'm going to draw in here is our hydrogen, because we want to reabsorb our hydrogen. That's going back to our blood supply, and we are taking in our potassium. And what happens is the potassium goes through a channel and that is secreted too. Okay, so just wanted to give you a little background on how we would control that there. This is going to be it for chapter 27. This screencast went longer than I wanted, but please reach out if you have any questions at all. And like I mentioned, you will have about 30 questions on chapter 27. Use the study guide and study well. Have a great spring break.